Hi guys, this is Anushka Upadhyay, and some of you might know me due to the very first credential mentioned over here. You can go through it by yourself, whatever written over here. I'll talk about what the session is about. So I'm going to hold a immunology PYQ session for the upcoming GATE exam, and before I begin with it, I'd like to show my gratitude to my immunology teacher and Deepshikha ma'am for making me eligible to hold this session. So the rules are quite simple. You'll pause, you'll read, you'll know your answer, and you'll resume because मैं नहीं रुकने वाली हूँ. ठीक है? So the very first question is match the entries in the group 1 with those with the group 2. In short, मिलान करें. तो group 1 की पहली एंट्री बोलती है J chain. And J chain is involved in multimerization of IgA and IgM. अब ये मुझे कैसे पता? क्योंकि मैंने ये डायग्राम देखा, which is depicted beside letter P. Here it's clearly shown that IgA dimer and IgM pentamer have a J chain which are helping it to multimerize. Next entry says serpin. The serpin is a shorter form for serine protease inhibitor, which is a C1 complement inhibitor. Now, what is a C1 complement inhibitor? It keeps a check on complement system कि ये खुद को affect ना करने लगे. How does it do so? It binds to the active site of serine proteases, make them inactive. So, when it makes them inactive, it assures the safety of the own body. Now, moving on to R. Entry R, which says beta-2 microglobulin, which is a component of MHC class 1. ये भी मुझे कैसे पता? क्योंकि मैंने ये diagram देखा, which is depicted beside R. Now here we can see there are two diagrams, class 1 molecule and class 2 molecule, and we can clearly see that class 2 molecule has a prominent beta chain while class 1 molecule has a red color का गोला, which is denoted as beta-2 microglobulin. इसको chain भी नहीं बोलते हैं. Also, it is not bound to transmembrane segment, which is quite of a relevant feature of it. Okay, and the next entry says Artemis. So beta-2 microglobulin matches with 2. Moving on to S, which says Artemis. Artemis is involved in VDJ recombination. Those who know about VDJ recombination must recall VDJ recombinase complex, which is involved in cleavage and processing of signal and coding joints. Like here, this blue block fragment and this red block fragment has to be joined. For that, this extra DNA, this loop DNA has to be cleaved, and ये काम कौन करेगा? ये काम Artemis करेगा. Fine? So, I think we're sorted with the question, and our answer is C. Moving on to the next question, we have correct combination ढूंढो question. So we'll go through the statements one by one. So the statement P says CD4 positive cells make contact with macrophages and stimulate their microbicidal activity. Take Q says CD4 positive cells make contact with B cells and stimulate them to differentiate into plasma cells. Now, CD4 positive cells, that is helper T cells, once they are awoken, they will further evoke several types of different cell proliferation. That is macrophages, B cells, CTLs. CTLs, that is cytotoxic T lymphocytes. Further, moving on to R. तो P और Q दोनों ठीक है, because P में macrophages involved है, B में B cells involved है, Q में, so both of them are perfect. R says CD8 positive cells make contact with B cells and stimulate them to differentiate into plasma cells. Now this is wrong, because CD8 positive cells means cytotoxic T lymphocytes, which are known for granzyme based apoptization of self-altered cells. Now, what are self-altered cells? The cells which host an endogenous antigen, like virus infected cell, like tumor cells, these are self-altered cells. और इनके apoptosis को carry out कराते हैं CD8 positive cells. सिर्फ ये नहीं कराते, there are certain other mechanism involved, but CD8 positive cells, that is cytotoxic T cells, are involved in this function. So we have our answer C, because statement S says CD8 cells, positive cells make contact with virus infected cells and kill them, which is perfect. Now whatever I explained is given in this slide, and we'll move on to the next question, which is a numerical type question. Now it says that heavy chain weighs 65 kilodalton and a light chain weighs 25 kilodaltons, and one antibody has 2 heavy chain and 2 light chains, and IgM is a pentamer like we just saw in the previous slides. So there will be 10 heavy chain and 10 light chains. Easing the calculation, we have weight of heavy chain, we know how many chains are there, we'll multiply it. Likewise, we have the weight of one light chain, we know how many chains are there, we'll multiply it and we'll sum up to get the approximate weight of IgM, that is 900,000 daltons. Okay, moving on to the next question. We have a question from antibody structure. What statement is? Again, सही combination ढूंढो. Okay. So statement P says limited proteolysis of rabbit IgG with enzyme pepsin generates 2 antigen binding reagents. Now this is a wrong statement. Okay. And an FC fragment. Again, it is a wrong statement. Why? Because when the pepsin digestion takes place, the antigen binding portion is remain intact. That is, you get a FAB2 portion. You won't get a FAB portion because वो बीच वाला डाइसल्फाइड ब्रिज नहीं टूटेगा. Statement Q says limited proteolysis of rabbit IgG with the enzyme papain generates a single bivalent antigen region FAB2 and peptide fragment. अच्छा, इन्होंने दिया है उल्टा. Pepsin में we get FAB2 and in case of papain we get FAB fragments which are 2 in number, not FAB2, 2 FAB. Quite confusing. So P and Q, both the statements are wrong and we have our answer, that is R and S. Now you can see it here in this diagram that pepsin digestion says you'll get a FAB2 portion, that is antigen binding portion, intact, plus it will fragmentize the FC portion. But in case of papain digestion, you'll get 2 FAB portions and a FC portion intact. But in case of mercaptoethanol reduction, ये पूरी एंटीबॉडी को तोड़ के रख देता है क्योंकि ये सारे डाइसल्फाइड ब्रिजेस तोड़ देता है. So we have our answer and we are happy with it. We'll move on to the next question. Okay. It says a newborn was accidentally given a drug that destroyed the thymus. Bichara. Which one of the following would be most likely outcome? Okay. Option A says lack of class 1 MHC molecule, B says inability to rearrange antigen receptor, C says inability to differentiate to mature T cells, and D says reduction in T independent number of B cells. Okay, so class 1 MHC molecule ka formation is completely genetic. It has nothing to do with thymus. Okay, and the option B says rearrangement of antigen receptor. So antigen processing and antigen presentation is a different phenomena which has again nothing to do with thymus. But we know The T cell maturation takes place in thymus. So option C would be perfect. And destruction of thymus would have no effect on number of B cells. So we have our answer C. Moving on to the next question, we have the question about opsonin. Which of the following immune system component can function as a opsonin? Now, opsonin and opsonization is a quite of a beautiful phenomena which I would show. Now, this is a hand-drawn diagram. कोई मेरा मजाक नहीं उड़ाएगा. Bear with that. So, whenever we have an antigen in the bloodstream, it will evoke a B cell proliferation, which will lead to further antibody production. And this antibody will go and bind to antigen. Now, this antibody antigen complex is there in the bloodstream, and this macrophage is also there in the bloodstream. Now, the thing is, macrophage have the receptor for FC portion of antibody, जो कि free है, क्योंकि binding तो FAB portion से होनी है ना, तो FC portion तो free है, ये free है, इसके पास इसके लिए receptor है, और जब ये बगल से गुजरेगा तो ये दोनों चिपक जाएंगे. And when the FC portion and the FC receptor binds, the macrophage will sense the presence of antigen, क्योंकि पास में है. In the closer vicinity, it can recognize better. So what antibody is doing here? It is increasing the chance of getting phagocytosed. So whatever molecule which increases the chance of an antigen to get phagocytosed is an opsonin, which is exactly what antibody is doing here. So we have our answer, antibody. Okay. So we'll move on. There are certain more opsonins, I forgot to add them here, but there are C proteins, there are MBL proteins which are also involved as opsonin in the process of opsonization. So the next question says the human MHC is, it's easy, it's polygenic and polymorphic. Why? Because there are a number of genes involved in MHC formation. To name a few, HLA1, HLA-A, HLA-B, and there are certain more involved in this MHC formation. And when there are so many genes involved, they are not going to make an identical product. They'll rearrange and make different products. That's why it's polymorphic. So this is sorted, and then moving to the next question, which says, which of the following condition promotes development of human autoimmune disorder? Basically, autoimmunity किस किस से हो सकती है? Which says, statement P, inability to eliminate self-reactive lymphocytes. When you cannot eliminate the self-reactive cells, it is for sure a kind of autoimmunity. So statement P is right. Q says generation of autoimmunity. This condition is also perfect because when autoantibodies are generated, we are having an autoimmune disorder. Statement R says ability to eliminate self-reactive T cells. Now, statement P and statement R are opposite, and one of them has to be true, and we know P सही है, तो R गलत हो गया. Statement S says induction of regulatory T cells in the thymus. Now, regulatory T cells are those cells which show intermediate affinity towards the antigen, and therefore they keep a check and help in prevention of autoimmunity. So, as scenario, it prevents autoimmunity. It do not conduct autoimmunity. So we know that the answer is P and Q and the answer is C. सबको लग रहा होगा C ज़्यादा आंसर्स हैं, नहीं? Okay. So the next question says, which one of the following is not true for class 1 MHC protein? Okay. So class 1 MHC protein are polymorphic. ये तो हम अभी पढ़ के आए, तो ये तो सही है. T cell receptor recognizes MHC class 1 protein. yes, it recognizes MSC class 1 protein as well as MSC class 2 protein. एंटीजन प्रेजेंटेशन के टाइम पे क्लास MHC ही प्रेजेंटेशन में होती है इन्वॉल्व. So this is a true statement. Statement C says MSC class 1 protein are displayed on the surface of nucleated vertebrate cell, which is right. This is perfect. All the nucleated cells do contain MHC class 1. Moving on to statement D. Says beta 2 macroglobulin is covalently attached with MSC class 1 protein. It's a wrong statement because it's microglobulin, not macroglobulin. So we are sorted with these two questions. Moving on to the next question. I think it's the last question also. Which one of the following statement is true with regard to processing and presentation of protein antigen? Okay. So in the class 2 MHC pathway, protein antigen in the cytosol are processed by proteasomes. ऐसा. Okay. Statement B says in class 1 MHC pathway, extracellular protein antigen are endocytosed into vessels and processed. Like I just said before, class 1 MHC is involved in endogenous recognition. Now this is the wrong statement. It is involved in endogenous protein antigen processing, class 1 MHC. So B statement is wrong. In the class 1 MHC pathway, transporter associated antigen protein processing protein, the TAP protein, is required for translocation of processed peptide generated in the cytosol. Yes, it's right. TAP proteins are involved in transportation of sub-processed antigen from cytosol to the endoplasmic reticulum. So this statement is right. D statement says invariant chain in endoplasmic reticulum is involved in transportation of peptides and loading of class 1 MHC. So the invariant chain ka concept is there in class 2 MHC. It has nothing to do with class 1 MHC. So the true statement would be C. Now you can see it in this pathway that the endogenous antigen are taken with TAP protein into the ear, like I just said. And in the endocytic pathway, proteasomes are not involved. It is involved over here. And in the exogenous antigen, which is involved in MHC class 2 complex. So the exogenous antigen are endocytosed and they are chopped into peptides and are loaded onto the MHC in case of class 2 MHC. In case of class 1 MHC, the endogenous antigens are proteasomally digested first in the cytoplasm, then they are carried to the endoplasmic reticulum via TAP protein where they are loaded onto the MHC. So I think we had a good session and I'd like to say thank you for your attention and tell us how you find it. Thank you very much, guys, for bearing me, of course.